Today we will discuss the final figure that is presented in this paper that we are discussing, where they have developed a custom designed PA-ECM model to culture the pancreatic cancer cells, and its characterization is more similar or more closely resembles with the in vivo pancreatic cancer tissue. So in this figure, what they have compared is that they have compared the drug response of the pancreatic cancer cells in different ex vivo model, mainly in the PA-ECM, that is their custom designed PA-ECM models, and in the organoid models. And they have compared the result of the drug response in three different kind of patients which has a different chemo resistance. And as we have discussed in our previous videos, that chemo resistance is mainly dependent upon the abundance amount of proteins. Is mainly one of the factor for this chemo resistance dependent upon is the abundance of the protein present in the uh, present in their pancreatic cancer cell tissues. And they have taken three different kind of populations which has a different chemo resistance effect. That is 1 2 5 5 6 has a higher has a least chemo resistance effect, and this 1 to 560 has a low intermediate chemo resistance effect, and 1 to 707 has a highest chemo. Oh, sorry, I have said the reverse order. Is 1 to 556 has the least chemo resistance effect as the tumor volume is higher, and the 1 to 560 has the highest has a intermediate chemo resistant effect, and 1 to 707 has least chemo resistance effect because the tumor volumes are lower, so chemo resistance is lower in this case. And the H&E stainings data are shown here for the intermediate chemo resistance populations, where we can see that when the pancreatic cancer cells that are cultured in this PA-ECM are treated with this gemcitabine and this NP drug, they can see that reduction in this pancreatic cancer cells, and also this proliferation marker, that is Ki-67, also decreases, and the apoptotic marker seems to be increased compared to the untreated cells that are not treated with the gemcitabine. And thereafter they checked this they quantified this Ki-67 and cleaved caspase 3 in all the three different populations, and what they find out is that with decrease in the chemo resistance, they see that increase in the apoptotic uh, signals, that is cleaved caspase 3, and decrease in the proliferation signals. Whereas for the least resistance, they see the decrease in the is apoptotic signals and increase in the proliferative signals. And this triple represents that triple culture, that is the PA-ECM also contain pancreatic stem cells and macrophages, so that will also contribute towards the chemo resistance. And in the literature, it has been known that one of the marker for the pancreatic cancer cells is this tetrasaccharide CA19-9. However, that marker approach does not work uniformly across all the samples, and the data about their CA19-9 biomarkers for the detection of pancreatic cancer cells is shown in the supplemental figures. So the better approach that they think is the better they used for the pancreatic cancer cells, so marker detection, is that intact G1 cells after the treatment with the drugs. And what they find out is that the PA-ECM reflects the drugs. PA-ECM reflects the G1 population similar to the in vivo results, whereas the other models such as organoid or spares represents the reverse results. And the data about their data where they compare in vivo and ex vivo G1 cell populations is shown in this panel E and F. Panel E is about the PA-ECM culture. So here we can see that for this 1 to 556 and 1 to 560 and 1 to 707 populations, there are some overlaps between G1 populations in all the three cases, and these overlaps are found to be similar to this intact G1 cell populations. That is, for this 12556, 40 around 38 to 35% populations are overlapped in the G1, and this seems to be correct from their flow cytometry, cell cycle analysis, and similar results are observed, seems to be correct for this 12560 and 12707. However, when they check for the organoid, they can see that the G1 populations in the in vivo seems to be very high, whereas when they check for the ex vivo results, they can see that the G1 populations are very low, and on the other hand G2 and M populations are very high. So this shows that the other models such as organoids does not show the proper representations or proper characterizations of the pancreatic cancer cells when they compared with the in vivo pancreatic cancer tissues, whereas PA-ECM shows the more better characterizations and resembles more closely to the in vivo pancreatic cancer cells. So from this figure they made a point, or they further substantiate their point, that their custom designed PA-ECM provides better in vivo characterizations compared to the other in vitro models or ex vivo models available. If you want to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.